Okay, welcome everybody, and thank you for taking the time to come and listen to my talk today. I am indeed going to explain a little bit about what kojoX is and how we're applying that manufacturing philosophy to advancing biotherapeutic manufacturing. So three themes I'm going to talk today a little bit about: a little introduction, a short introduction to Fujifilm biotechnologies. And then I'm going to talk a little bit about what we mean by kojoX and how we're using kojoX as a philosophy to really drive agility in our supply chains. And then I'm going to talk a little about some new technology that we've brought on our stand here today with some of the team, where we're really trying to disrupt the paradigm on how we do biomanufacturing. So quite a lot to cover in the time, but we'll get through the content and then have some time at the end for questions, hopefully. So we have at Fujifilm Biotechnologies a Partners for Life strategy. So we operate as a contract development and manufacturing organization, sitting with our partners who would do the research and development, manufacturing, developing, scaling through to commercial manufacture, to ultimately drive medicines through to patients. And we underpin that Partners for Life strategy with three themes: putting our people first, which we believe is actually key to all of this, the way that we operate our business; utilizing technology and science and innovation to transform the industry. And I'll give you an example today through continuous manufacturing, how we're trying to do that. But ultimately, as a service provider, unprecedented delivery, that's absolutely key to being a successful CDMO and to have a key partnership. So just a little bit of context: we are part of Fujifilm, the holdings company, obviously from Japan, 90 years old as an organization. And actually for many of those years, they've been in the healthcare space. Historically was known as that camera company. But actually, over sort of 15 years ago now, recognized with digital cameras coming along, which they were developing themselves, they were disrupting that market that they dominated through selling films. So since then, really pivoting towards being a healthcare organization. And you can see some of the numbers at the bottom. Actually, about a third of the global revenue for Fujifilm is from the healthcare business. And as we continue to grow and continue to invest, we recognize a higher proportion of that growth in the future. So we've been doing this for a long time, and actually sort of strong data knowledge and AI has been at the heart of this. 20 years ago, when you were taking a photograph and the red eye was being removed from those photos, that was AI doing that before we even knew to call it AI. So, for a long time, recognizing how we can leverage that has been really key for us. And today we're deploying AI in all sorts of different areas: the way that we design experiments, the way that we're developing next generation inline filters and sensors, and even how we're managing our deviations in a GMP quality environment. So we're really trying to not just talk about AI, because lots of people like to talk about AI, but actually how we're going to utilize it in today's technologies and tomorrow's. We're an organization based on innovation, and actually over 55,000 patents now as part of that Fujifilm organization. And in the last 10 or 15 years, a proportion of them coming from that life science space. So this is a little bit about the context of our wider organization. And specifically within the Fujifilm Life Sciences organization, this is a group of companies within that holdings company, all represented on the booth here today. We have a range of different services and capabilities. We at Fujifilm Biotechnologies are providing CDMO services across a different range of modalities: cell culture, mammalian, microbial, advanced therapies, and beyond. So really operating in that space between, on the left-hand side of discovery and the right-hand side, that patient. And we have a global network and a growing network. And actually, if we talk about some of the geopolitical situation that we have today, having that presence in Asia, in Europe, and in the US, we feel is a real strength. And we're continuing to roll out development of our kojoX manufacturing facility, having those capabilities across that network. We don't have every single capability in every network because we want to play to the strengths and the heritage of those individual locations. But as you can see here, we're starting to roll out that capability so that you have flexibility in your supply chain depending on what the current geopolitical challenges of the day. So we talked a little bit about kojoX, and I want to spend a little bit more time just to try and unpick that and what we mean by kojoX and how we're bringing agility to our supply chain on your behalf. So in a sense, kojoX is the way that we design our facilities. We're talking about standardizing. We're talking about an operating philosophy around modularity. We're talking about being able to go into a manufacturing facility in Europe and having all of the same equipment, all the same design layout, the same quality management system, the same way of operating, exactly the same as, say, a facility in the US or a facility in Asia. We've already started to deploy this kojoX facility with our 20,000 litre manufacturing suites. So the Holly Springs facility that we only opened a couple of weeks ago is really a carbon copy of our manufacturing facility in Denmark. So actually from an inside, the operator would not know which of those two facilities they're in, because it's exactly the same equipment with the same systems, the same automation and what have you. And that then starts to give us real advantages when we talk about moving programs around the network. And already with some of our partners today, we're talking about running the process in one facility and actually being able to run it in another facility on another site without having to repeat PPQ. And then you start to think about speed in tech transfer. Then you start to think about taking risk out and cost out and ultimately have agility in that way. So there's a range of different ways in which we're trying to standardize, and we've deployed that kojoX philosophy at our 20,000 litre facility. But we're also trying to deploy that in our smaller scale facilities. So later on in the presentation today, I'll talk a little bit about our first single-use kojoX facility that we'll be bringing online in the UK in the first quarter of 2026. So that's taking us up to 2K and to 5K, fed batch and continuous in our first single-use kojoX facility. And as we continue to deploy those kojoX facilities across Asia, Europe and the US, again, that same consistency, the same equipment, the same ways of working, the same operating philosophies. So again, operators and customers wouldn't know the difference between which facility they're in. And here's just a little bit of a schematic as to how we anticipate being a partner for life. We start the journey quite often in an early phase preclinical environment, and we'll go through process development. Within our network, we have about 500 process development scientists developing processes across mammalian, microbial, and advanced therapy capabilities, cell line development capabilities, all the way through to late phase product characterization and process characterization. And as the molecule works its way through the journey, we then have options as to where and what scale we deploy the process, depending on market needs, the customer needs, the patient needs, and the disease state. So in some cases, we're scaling up from lab scale to 2K, to 5K, to 20K. In other cases, we may scale out, so you may get to the 2K and then just have multiple batches at that 2K or 5K. Or in some cases, we have a new concept called scaling on, and that's actually porting the process into a continuous manufacturing environment. You may want to do that because of product stability issues. You may want to do that for some different reasons, and we'll get to some of the drivers for that. But what we're trying to do is to create an ecosystem for being a partner for life. So every stage from that early phase through to commercialization, and then ultimately scale down again at the end of the product life cycle, we've got the right capabilities in the right geographies for your needs. So we've talked a little bit about Fujifilm Biotechnologies. We've talked a little bit about our kojoX philosophy, which we feel is quite unique in the industry today as a way of growing. We've taken that conscious decision not to just acquire sites, and then as you acquire a site as a CDMO, you're stuck with the automation system that site has. You're stuck with the equipment that site has. So our growth and a significant growth journey that we're on is through that organic purpose by design. So staying with that standardized design, the discipline that's required of an organization, not to just allow facility designers and engineers and operators to sort of change things because it makes sense in a local place. By keeping with that kojoX philosophy, that's how we're trying to drive that differentiation within the industry. And now I'm going to talk a little bit about an example of some of the innovation that we're doing within Fujifilm Biotechnologies and how we're trying to differentiate ourselves through that innovation. We have a toolbox for advancing tomorrow's medicines, and we have a range of different services, capabilities, science, technology, and products to do that. And I'm going to give a little case study here around continuous manufacturing and take 3 of the elements out of some of the toolbox here as part of that storytelling. going to talk a little bit about kojoX, which is our proprietary CHO cell line system. Many of our customers come to us with just a gene sequence, so their tech transfer is pretty quick, and they utilize our kojoX cell line for their needs. Of course, we operate and work with a lot of our large pharma partners where they have their own cell lines, and that's fine as well. We can work with those. I'm going to talk today a little bit about a technology called Symphonix, which is a downstream bioprocess equipment that we designed with an end user in mind. So we, over the last 25 years of being a CDMO, we've ran many different processes, different scales, different ways of working, and we've recognized some of the needs that we needed from our bioprocess equipment. So we actually embarked on a journey about 7 years ago now in designing and developing and now deploying that Symphonix technology within our facilities. And then I'm going to talk about MaroX, and MaroX is our continuous manufacturing service using our kojoX cell line that we adapted for both fed batch and perfusion, using our Symphonix systems, which we've designed specifically with an ability to run in a continuous mode, and pulling that all together in a case study. So a little bit on the cell line. We've been operating and evolving this cell line for about 10 years now. We're continuously and aggressively developing and making sure that this cell line is cutting edge. Approximate titers of about 5 to 7 grams per liter, although we have had a 10 gram per liter as a sort of a headline there for the right particular molecule. And we've demonstrated our kojoX cell line, not just for MABs, but for some of the more complex molecules, some bispecifics, now some trispecifics, and some fusion products as well. So quite a broad and adaptable cell line. More recently, we developed and evolved our kojoX cell line so it could be used in both a continuous and a fed batch environment. Some of our partners in the continuous space start their journey in fed batch, and maybe after a certain phase, because the regulatory environment allows you to do this now, port across to continuous, maybe at late phase before commercialization. And by doing that, by having one cell line that they can jump between those two modes of manufacturing, is one less variable from a regulatory perspective. So we embarked on a journey of evolving the kojoX cell line for perfusion-based organizations. We then talk a little bit about Symphonix, and we started to develop the journey for Symphonix, as I mentioned, about 7 years ago. We've been deploying it in a GMP environment for about the last 4.5 years, in a range of different modalities and a range of different scales. And most recently, we actually launched a larger version of the Symphonix, the image that you can see on the right-hand side of the screen there. And we have one of these systems on the Fujifilm booth. So please come along. Some of the team in the room here, but they'll be very happy to talk you through and give you a bit of an overview of the technology. But what we're looking at here is a new way of operating downstream bioprocessing. That downstream bioprocessing has not really had that much innovation over the last 25 years. We're still running the same chromatography that we were doing 25 years ago. Yes, the systems have evolved, the pumps have got better control, the resins are better, but actually fundamentally the downstream process has not really changed. So we embarked on this journey to really disrupt that downstream process. In the UK site, we opened the very first single-use manufacturing facility for MCC in Europe in 2013. And if you go back to 2013, you would see lots of different equipment for the downstream pieces of equipment. You'd see a chromatography skid, you'd see a TFF skid, you'd see a viral inactivation skid, and all of the different unit operations required for downstream processing. Every one of those bits of equipment generally came from a different supplier. It was single use, so they generally had a different flow path. Because it was from a different supplier, it had a different operating software, and all of our engineers then had to spend time qualifying and maintaining different pieces of equipment. So you can imagine from a CDMO and operations perspective, significant complexity in our operations. So we embarked on a journey of designing a new bioprocess equipment, one piece of equipment that could do all of those unit operations. So on the left-hand side there, this is a chromatography skid. It's also a viral inactivation skid. It's also a TFF skid. All of the requirements required for downstream processing in one unit operation. This means one piece of hardware for our equipment teams to maintain. It means one piece of operating software for our operators to learn. It means one flow path. So our supply chain becomes a lot easier, and we don't have a warehouse filled with lots of different flow paths from different suppliers and different scales. We launched the system on the left-hand side there into our GMP facilities about 4 years ago. So we've really been trying to build that experience of in a fed batch mode. Fully single use from using all of the same requirements as any single use flow path extractables and leachables and all of the things that we need to ensure patient safety. We also recognise thinking forward, and again looking at how downstream has been operated for the last 20 years, very little disruption in buffer management. And actually, as we see process intensification going forward, and we see the importance of sustainability and the way that we're designing and operating bioprocessing today, advancing buffer management was a real requirement for our equipment. So we integrated into the Symphony-X inline dilution and inline conditioning. So you could plug your normal buffers in like you've done for the last 25 years and just run those processes in the normal way. But you could also plug in buffer concentrates, up to 20 times stock buffers, which would be diluted at the point of use. So your footprint for buffer makeup and storage pre, during, and post-use reduces significantly. It also has inline conditioning capabilities, so you can plug an acid, a base, a salt, and a water, and really do at-point dilution just before it goes on onto the unit operation. Then we're starting to really move the dial on the advancing buffer management. And finally, we recognize the need to evolve the automation. The equipment here on the left-hand side and the right-hand side is designed for fed batch operations. It can run a chromatography skid, it can run a TFF mode, all in the same hardware, flow path, and software. But we also designed it so that Symphony-X could be connected together. We could start to connect multiple Symphony-X's together through the integrated automation software, so we could go towards continuous and connected bioprocessing. So now we have one piece of equipment that can do fed batch and continuous. Again, a real disruption in the industry, because today, if you want to design a continuous facility, you need to design it and you need to fill it with continuous equipment. It can't really be utilized for fed batch processes. But with Symphony-X, we can now disrupt the way that we design and operate our manufacturing facilities with one process facility that can run fed batch one day and it can run continuous the next day. Really trying to design kojoX supply chain agility into the way that we're designing it. And roughly on scales, the system on the left-hand side is pretty much set up to service a 2,000-litre bioreactor. The system on the right-hand side is really set up to service a 5,000-litre bioreactor and beyond. So that's sort of the scales that we deployed here, and these reflect the facilities that we're designing today. So we took this paradigm of being able to scale up from 2K to 5K to 20K. We took this paradigm of being able to scale out, and there's sometimes there's a need for that as well. So get up to 2K and then just more batches. But then we use this term of scaling on and moving into a continuous manufacturing place. We've demonstrated some good cost of goods reductions in there. And what we're actually seeing now from the industry is molecules that previously were discovered looked like they had great drug efficacy, but because they didn't have the good stability in terms of being able to be expressed in a fed batch bioreactor, those molecules were discredited for development. Now you can go to a more competitive perfusion expression system. Now you can go to a connected and integrated downstream through Symphony-X, and you can start to redevelop now drugs that were good efficacy but not very good product stability in a perfusion environment and drive a different way of considering molecule design. And a lot of people, and we were part of that community over the last 7 or 8 years, talked about continuous manufacturing. There was a lot of dialogue with the regulators. There were white papers issued from the FDA, and we were actively, vocal and present in those communities, looking at how we would be able to really, truly bring continuous to the downstream space and to the mammalian cell culture space. And there was a lot of talk, but there was not a lot of scale-up data to demonstrate some of these principles. So we embarked on quite a significant innovation program where we demonstrated a 500-litre perfusion scale, this concept. We took our Apollo X cell line, which we'd adopted for both perfusion and fed batch operations. We took a batch strategy that we'd developed and evolved in collaboration with the regulators to ensure that this would be appropriate from a QP perspective, from a quality perspective, and a way to schedule. We took our Symphony-X systems, which were designed to be connected together for integrated bioprocessing. We took our already well-established MAB platform and adapted that to that continuous manufacturing facility. And then we also collaborated with one of the other Fujifilm life science organization companies, formerly known as Irvine, to adapt some cell culture media for continuous manufacturing. We knew one of the challenges historically with continuous was the cost of the media. Every single day in a perfusion bioreactor, you're effectively replacing all of the media. So a 500-litre bioreactor ran for 10 days, 5,000 litres of media. So we needed to drive down the cost of goods of that media to make this approach viable. And then we took some quite innovative approaches to how to really drive cell density to allow us to operate in this perfusion culture, pull it all together into a proof of concept run. This is effectively a high-level process flow of what we looked at here. We effectively took that show perfusion-ready cell line through a wave perfusion into a perfusion bioreactor. In our proof of concept lab, that was a 500-litre perfusion bioreactor, through a cell retention device into these connected and integrated Symphony systems. We took that and we used that with the buffer concentrate concept that we talked around. So instead of having lots and lots of one times buffer filling the clean room space, we had buffer concentrates diluted at the point of use, sourced through a GMP integrated supply chain to pull all of this proof of concept together. And we were really pleased with how this worked out. There's just an image at the bottom here of those 7 Symphony systems connected together. I've used this internally. I'm a downstream guy, so I cut the photograph of the perfusion bioreactor off on the left-hand side, but there was a 500-litre perfusion driving out there. And just in the background, in the blue there, you can also see the buffer concentrates that we source, plugged into the Symphony, diluted at the point of use. And there's some data here to show that this was a scalable process. We ran the process at 50 litres perfusion. We ran it at 500-litre perfusion with the same product, tighter. We demonstrated here in the middle graph, same product quality. So perfusion wasn't going to have a decremental impact on the quality of the product. And in fact, because the perfusion environment is a much more gentle environment for expressing and operating with the cells, you tend to actually see better product quality in a perfusion environment. The drivers for perfusion when they were first conceived years ago in microbial. And we also demonstrated through the connected downstream residual clearance of HCPs, DNA, and all the things that you need to ensure product quality and product safety. We've published this data, and there's a scan here to a biotechnology progress publication, so people can see this in a peer-reviewed journal to just ensure that we had that external validation, that the science and the innovation and the way that we were transforming the industry through that was published in an open environment. This was just a concept here to show how we took that buffer management, and actually we reduced the footprint of the buffers in those clean rooms by about 50%, a little bit more than 50%. Since doing this, we've continued to evolve this technology even further, and we're at the point now where we can use buffer concentrates, inline condition, and deliver a gradient through that condition, through the control systems of the Symphony. So a way for us to drive footprint reduction in our clean rooms. This is going to drive us towards more sustainable operations. We don't have large clean rooms with lots of HVAC costs, and advancing the way that we're intensifying our processes. And this was just an example in our non-GMP proof of concept facility that we developed. The process was generated in a fully automated way, about 1 kilogram of antibodies every single day. So now you start to think about that scale-on philosophy. If you need 5 kilos of antibody, you run it for 5 days. If you need 50 kilos of antibody, you scale it by time, as opposed to transferring the process from one facility to another. So the facility you start in your early phase, maybe you run it for a week to get enough material for the early phase clinic. It's exactly that same facility that you'll run for your late phase, your PPQ, and your commercial manufacture by just scaling by time. We demonstrated here at a 500-litre scale, but our new facilities that we're about to bring online has up to 5,000-litre scale. There's some technical challenges at that scale for cell retention, but in principle, you could think about running a perfusion bioreactor at a 5,000-litre scale every single day, generating hundreds of kilos of antibody. So real ways to differentiate the approach to biomanufacturing. And this is our facility that we're bringing online in the UK. We're really excited about this facility because this is the first kojoX facility for our single-use biomanufacturing. just as described earlier in the talk, where we took all of the operating philosophies from Denmark at the 20K, and we copied and pasted that across into our Holly Springs facility. We're using the facility in the UK here to redefine what we mean by single-use kojoX. So as we start to bring facilities online in Asia, and we start to bring facilities online in the US, we've got exactly that same consistent design and operating philosophy, equipment list, qualification list. So we really start to embed agility in our supply chain. When this facility is completely full and up and running, it will be the largest single-use biomanufacturing facility in Europe. The first of the 5 lanes will be bringing online in the first quarter of 2026. And I was walking around the facility last week, and equipment's delivered. The clean rooms are looking really good. We're starting to test water systems, and our ability to expand and activate future lanes is really quite quick because of that kojoX facility. So really excited to bring that facility online. So that's the end of my talk today. I wanted to just sort of summarize this concept of thinking about how we're trying to disrupt the industry through innovation. We're utilizing our kojoX facility to drive agility through our supply chain, and ultimately then bringing all of those capabilities to you as a need in an industry as to be a partner for life.